everybody, and I don't know, she said she didn't want to, Chris Simonello is actually the official host, but she said she didn't want to introduce Kiho because she was afraid she'd tell too many stories.
Thank you.
And apparently she has a few to tell. And a reminder that there is a potluck get-together at Chris's on Saturday evening. If you would like to come and socialize and get away from, Chris lives a little bit further away from the noise, right? Yeah. And so Dr. Kim is a professor in the Department of Environmental Science at American University in D.C., and he earned his biology degree from, his BS in Biology and Environmental Studies from Brock University. Where's Brock University?
St. Catharines, Ontario, Canada.
And he earned his master's degree at Florida International, down the road, a little closer to our Florida reefs than we are. And I guess that's where he connected up with Chris. And he earned his PhD in biology from the University of Buffalo. He's a marine ecologist. He works on a variety of things that Jean Shin and I have been working with for many years. Nutrient dynamics in reef environments. Most of you don't know, there's only a couple people, 3 or 4 people in the room that know that I was known in the 80s as that crazy lady who thinks that nutrients aren't good for reefs. You know, fertilize them, make them grow better. You know? And we've been doing experiments to do that. He also is, Dr. Kim is also interested in the octocorals and the aspergillosis problem in sea fans, which Jean has been interested in for many, many years. And so, coral diseases, octocoral diseases. In fact, he's got a couple of chapters in the new book on diseases of corals. And his talk today is nitrogen dynamics in tropical coastal ecosystems, a case study in Guam. So he's a man of many talents.
Well, thank you. Thank you very much. Thank you, Chris, for inviting me, and thank you for hosting my visit here. So the focus of the talk that I'm going to be giving today is about the importance of nitrogen in tropical coastal ecosystems. And although the talk will end in Guam, I'm going to actually start a little bit about the work that I did in the Florida Keys to give you some context and a route for why I ended up in Guam to begin with. And before I do that, though, like all coral reef talks, it has to begin with a gloom and doom, and so here's my contribution. And all of you know that these reefs are declining for lots of different reasons. On the Great Barrier Reef, this recent paper in PNAS, they talked about the importance of cyclones as well as the outbreak of the predator Acanthasida, the crown of thorn. Whereas in the Caribbean, disease has played a very significant role, particularly in the last 30 years or so, with the demise of the aquaporids as a result of a disease outbreak in the early 1980s. And so there's a combination of things that are going on. And so there has been some discussions about which factors are especially important. Where I sort of jumped into this discussion was a paper where we discussed the variety of things that were happening and the possibility that diseases could be an important driver of ecological change on coral reefs. When we began this work back in 1999, there may have been about 3 or 4 diseases. Up today, there may be 40 or so diseases or conditions or signs that afflict both Caribbean and Indo-Pacific corals. And so there's been sort of an exponential growth in the number of afflictions, disease-related infectious diseases that cause demise in many of these corals. This is a sea fan, Gorgonia ventolina, that is very common in the Florida Keys. And while we're working on this paper, there was a disease outbreak called aspergillosis. Aspergillosis is a fungal pathogen that we think came from land. Where exactly on land it came from, that's still up to dispute. Some think that it's a locally introduced fungal pathogen. Some people think that it may have been carried over with dust from North Africa. That hasn't been resolved yet, but around 1997-ish, this disease broke out in the Caribbean. Within a period of about a year and a half, it has spread from southern part of the Caribbean Sea to the northern part, including the Florida Keys. And so we began monitoring this disease outbreak to look at the impacts of disease in the Florida Keys. Now, as you know, in the Florida Keys, hard corals are largely gone, and so these octocorals are really the dominant players, both in providing habitat and also complexity to many of the reefs along the Florida Keys reef tract. And so we carried out a long-term monitoring project, and what we find is, like with many diseases, there is a peak and eventually it dies out. And so these are different parts of the Florida Keys, the upper, middle, and lower. This is where you want to look at because this is the average, and so he peaks and eventually declines. And by the end of that 6-year period, the disease had become endemic and largely had disappeared from the population. If you look at the bottom panel, which is disease severity, that is the amount of tissue affected by the outbreak, that has stayed the same. So what's really happening is the number of sea fans infected is really low, but those who are infected still have signs of this particular disease. I went back in 2008, and by that point, the disease had really become endemic and occurred at very low frequencies. And so we think what was happening was that over the course of the 6 or 7 years of the outbreak, there was a turnover of the susceptibles. And so what we now have is a group of sea fans that are now resistant to the fungal infection, because we can't imagine that the fungal pathogen has somehow disappeared from the aquatic environment. So one of the questions that came from this sort of work was, well, what is causing all of this variability, both in terms of the severity of the disease as well as the disease prevalence? And so one obvious sort of large sort of factor is the role of temperature. And climate change and related impacts on ocean acidification, those sorts of things. So these large sort of global scale drivers are one source of variation for the disease outbreak that we saw. The alternate is more locally based kinds of explanation that has to do with the input of sewage, specifically land-based nitrogen pollution that's washing into the ocean. Many reefs developed in very oligotrophic waters, and so the addition of lots of nutrients may be also an important driver of ecological change. Now, as we'll see, I think, and I think a few other people in this room, think that nitrogen is probably the more important, at least at the local level. Clearly, there are reefs out there that are still doing well, they're still thriving, and so that seems to point to local causes of ecological change. Now, if you think about nitrogen versus, say, CO2, nitrogen availability has doubled over the last century with our ability to make synthetic agricultural fertilizer. That's a huge change in the nitrogen cycle. During that same time over the last century, our population has grown from like 2 billion to about 7 billion, talking about lots of human-derived sewage that has to go somewhere. In many places, especially around these tropical coral reefs, they're ending up in the ocean. And so we began to think about whether or not we can correlate our data with the sea fans with water quality data. And lucky for us, as you all know, the Florida Keys is incredibly rich, relatively speaking, in terms of environmental data. And so the sites that we chose for the sea fan monitoring also coincided with many of the sites that were being monitored by the Southeast Research in the Water Quality Monitoring Project. And so we looked at lots of different parameters, and I can tell you right off the bat, temperature had no relationship with disease. Now, where we did find a relationship was between disease severity and the availability of nitrogen as a function of phosphorus, some ratio between nitrogen and phosphorus. Now, this result is not completely surprising because there have been other experiments that show that if you add nitrogen as well as phosphorus. So John Bruno at the University of North Carolina, Chapel Hill, he took a little packet of Osmocote, which is a slow-release fertilizer, and he pretty much stuck it onto an infected sea fan and other corals, and they noticed that with the addition of nitrogen and phosphorus, the size of the wound increases over time versus controls. And so we know that there's some linkage between between fertilizer, nitrogen in particular, and disease dynamic. And a couple of other people have shown very similar kinds of trends. And so this was really sort of interesting. We also then asked the question, where is this nitrogen coming from? And one of the ways you can look at that is by using stable isotope analysis. And so we, for the same sea fans that we've been monitoring, we clipped a little piece off and carried out nitrogen stable isotope analysis. And the sites are arranged from top of the keys to the bottom of the keys. And there's a slight increase in the nitrogen signal, the del N15 signature, which indicates that as you go from the northern part to the southern part, sewage is more dominant than, say, synthetic fertilizer, which are probably the two most dominant sources of nitrogen in this particular area. As you may know, synthetic fertilizer has a del 15 signature of around 0, whereas sewage can be anywhere from 8 to 15 to 16. And so they sort of mix and match, and so some combination of the two can result in some median value or some middle value. These corals, these sea fans, if you pick one out of, say, the Bahamas, which is relatively pristine in terms of sewage impact, their isotopic values for nitrogen is somewhere in the order of 1. And so we know that these sea fans in the Florida Keys at least are being impacted, or at least picking up the signature of land-based, primarily sewage-based nitrogen, and incorporating them into its tissue. But we do know that these values do reflect combination of fertilizer and sewage, and we also know that because of the production of synthetic fertilizer, there's been a shift over time, and so we wanted to ask the question, what is the dominant source of nitrogen for not just the Florida Keys, but for the Caribbean, and has that changed over time? We sort of had the guess that a long time ago, when fertilizer wasn't that important, it would be dominated by sewage, and over time as synthetic fertilizer, and many of the islands began agriculture, the signature would then shift from high N15 values to low N15 values, reflecting sewage versus fertilizer. And then in some places there's an uptick as fertilizer is no longer a economically viable option, and many of the islands are turning to tourism, which would then bring back the sewage as a dominant source of nitrogen. And so being in Washington, D.C., we had the opportunity to rifle through all of the samples at the Smithsonian Institution and the collections of sea fans and other gorgonians that they've been collecting since the late 1800s. And so it was pretty amazing to be able to open up all those drawers throughout the collection and then pick out, and luckily for us, we just needed very small pieces of this thing, and so they didn't really have any problems without doing so. And so we clipped off a bunch of pieces and carried out the stable isotope analysis of these corals. And so on the top is Pseudopterogorgia. These are the sea feathers, sometimes they call them, or sea whips. And this is Gorgonia. We mixed a couple of species of Pseudopterogorgias and also the Gorgonias. There's Ventolina, and then there's also Flabellum. They're very similar. We know that because we actually carried out the isotope analysis and showed that they're not different significantly in terms of their isotopic signatures. And so on the bottom axis is time, 1840 to, well, we didn't go into the future, but it cuts off at 2005-ish. And so in both cases, what we see, generally there's a decline in the N15 signature, which indicates that the Caribbean as a system began with sewage as being the dominant source of nitrogen and shifting towards fertilizer. And so this is kind of cool. And if you split the data up between pre-1960 and post-1960, which reflects more or less the Green Revolution, we see a very dramatic difference in the N15 values. And so this is before 1960, mostly sewage. After 1960, primarily fertilizer. Now that's a very simple take on a very, very complicated system, because we know that fertilizer and sewage aren't the only contributors to the nitrogen pool in an aquatic ecosystem. There's deposition from fossil fuel burning, from volatilization off agricultural fields. There's upwelling. Lots of different things come into play, although fertilizer and sewage tend to be the dominant end members in the nitrogen pool. And so this study really sort of pushed me to look for a system that was a lot simpler, where the dominant source could be identified reasonably easily, and which would allow us to use the N15 signature not only to discern the source of the nitrogen, but also the intensity of the input, right? If there's only a single source of nitrogen, then the isotope values can say not only where it's coming from, but how much of it is entering that particular ecosystem. Now, the isotope work is, in many ways, I won't say better, but very different than, say, taking a snapshot with a water quality kind of a measurement, because isotopic analysis allows you to take measurements of a piece of tissue that has been integrating the nitrogen signature for various periods of time. And so if you, for example, if you take a piece of chlorpa or other pieces of fleshy macroalgae, you may see the nitrogen pool for the last couple of weeks, because that's how quickly they turn over the tissue. If you look at the corals, you may be looking at the nitrogen environment for an entire year, because that's how long maybe a centimeter of coral tissue takes to grow. And so picking different species for isotope analysis allows you to open different size windows into the nitrogen pool in an ecosystem. So one of the places that I ended up in terms of identifying a much simpler nitrogen dynamical system was Guam. Guam is in the middle of nowhere, and so the kinds of sources of nitrogen is very, very limited. This is a close-up because you can't really see Guam until they get really close. And there it is. From north to south, it's about 25 miles, so it's pretty short. And from this side of the hip to the other hip is about 6 miles, and so it's pretty manageable. And it's not particularly populous, although there is some substantial population. And you can see where the light areas are. That's where the population is mostly. Guam is sort of interesting geologically, because around here is a border between a carbonate platform in the north and a volcanic geography in the south. And the south is dominated by rivers, which empty out into these areas, whereas in the northern part, most of the water either runs off immediately or percolates through the carbonate system and then discharges to the surrounding waters through submarine groundwater discharges. It turns out that the rate at which water moves from rivers into the ocean is about the same as the rate at which water infiltrates the carbonate system and then out into the ocean water. And so in many ways, the groundwater discharges, submarine groundwater discharges act like rivers. So the island has about 160,000 people, but that population is dominated by or overwhelmed by tourists, which number a little over 1 million each year. About 75% come from Japan, about 10% from Korea, and the remainder from Russia and China. So it's a tourism-based economy, although the Department of Defense also contributes substantially to its economy. The island of Guam supports a number of Air Force and naval bases, and it's going to be more concentrated in terms of the U.S. presence because they are realigning the Pacific Theater and moving Marines from Okinawa to Guam over the next few years. And that's going to portend a huge increase in the population and intensity of activity on the island. The other sort of interesting thing is that there's virtually currently no agriculture. Almost all of the food, all of the produce that is consumed on the island is delivered from Hawaii. And so we don't have to worry about synthetic fertilizer. There are a couple of golf courses, but we're not talking about something that is huge relative to the population and the sewage that they would produce annually. They really have poor wastewater infrastructure, so they don't do tertiary wastewater treatment. So they have a secondary wastewater treatment plant, but only about half the resident population sits on a sewer line. The remaining half have septic tanks or open pits, and so the movement of sewage is relatively rapid and very directly connected to what happens along the shore. The corals reside on these platforms outlined in lighter blue, and it's a consolidated carbonate platform. And so you can see that onshore to offshore is relatively short. Most of the seagrasses that I'll talk about is around here. Corals occur all the way around the island, including the top part, but not too much on this side. This is the dominant direction of the currents. And the currents also flow south this way and then either split up that way or curl down this way. Some pictures of what the reefs look like in Guam. Very different than Florida Keys. So for that's a scale. For the person serving as a scale, these are large sort of Volkswagen-sized bombings of corals. The diversity in Guam is actually pretty good, and so you can go and find places where corals are pretty abundant. Lots of aquaporids. There are parts of the reef where it's completely dominated by these alcyonnaceans, these soft corals. This is Signularia. There are also small patches of mangroves, Rhizophora, as well as Avicennia. That is not a proof of life picture, by the way. She's actually she's actually happy to be there. And these are the seagrasses. The seagrasses in Guam is dominated by this really large, some of them are maybe a meter in length. This is Inhalus acroides. And they occur in large patches which become exposed during low tide. And so in many ways, it's a really nice place to work because you can simply walk out with your Home Depot bucket and just plop things down, take cores, sample, manipulate whatever you need to do, and then walk back to shore. In many cases, you don't even need to use your snorkel because you're just walking around, which the parents of the students whom I take really appreciate. As I mentioned before, the population density is spread out mostly along the northwest coast, the leeward side of the island. Most of the tourists are here, and so are the residents. The southern island, as you can see, lots of rivers occur there, whereas there are no rivers in the northern half because, again, it's a carbonate platform. So one of the sites, these are a variety of sites. This site here is Agania, is one of the sites that we'll look at, and there's another site up there that we'll look at. The stars indicate wastewater treatment plants. Again, all of them are barely operating at secondary treatment levels. Guam has been sued numerous times by the EPA because their sewage wastewater infrastructure is below requirements. So one of the first things that we wanted to do was, again, test the hypothesis linking nitrogen input and coral diseases. And Guam, like anywhere else, has its share of coral diseases, including white spot syndrome affecting this large peridid coral. And so we went out, and we didn't sample the corals because these corals don't have a lot of tissue. And so if you have to do nitrogen stable isotope analysis, you don't want to work with hard corals. You want to work with things around it. And so we picked Signularia, the alcyonnacean, because they co-occur, and you don't need a permit for soft corals, which is even better. And then you can sample them with a pair of scissors as opposed to a chisel. And so for lots of different reasons, work on hard corals. And so we found that if you look at the changes in diseases, so we sampled once every 2 months across a year. So going back, looking at the same transects of corals, clipping off little branchlets of the soft corals, and found that the 15N value was a reasonable predictor of changes in the disease severity of white syndrome on these large corals. So we know that there's now this link. One of the factors driving this link appears to be precipitation. Precipitation, indicated by rainfall in millimeters per month, has a interesting relationship with delta 15N values. We had one outlier there, which really drove me crazy, but that was because of a typhoon that passed north of the island, and that really kicked up stuff without adding a lot of rain, because it was beyond the rain part, but it was enough to throw the water up and churn things up and probably mess up the nitrogen signature. And so typhoons there was actually one time when there was a typhoon and an earthquake in the same day while I was on Guam. So, interesting place. So in addition to corals, we were interested in whether or not other coastal ecosystems were affected. Seagrasses was the other focal species, and so we did very similar kinds of things. We looked at isotope signatures, we did growth rates over a couple of 3-year period, resampling same transects and quadrats. And what we find is that these seagrasses vary in terms of their delta 15N signatures. They also vary in carbon. I'm not going to talk about carbon, but it says something about seasonal variation. And from year to year, there's not a lot as an entire island, but there from site to site, there are significant variation. Right? So So the question is, what's driving the site-to-site variation? Right? It turns out that there are a couple of things that drive site-to-site variation. One is population density that is adjacent to the seagrass beds. Now, these seagrass beds, as I said, you go to the beach, you walk out 5 steps, you're there. You're right on the seagrasses in many places. And so there's a very direct connection between what's happening on land and what happens on the seagrass beds. And so there's a very strong relationship between population density and seagrass delta 15, and also distance from input. And so we know there are lots of rivers, particularly in the southern part of the island, and so we can map how far a particular bed of seagrass is to the mouth of the river, and that is also a good predictor. And so farther away from a river you are as the seagrass, the less exposed you are to sewage. And if you look at our seagrasses, and when I say our, I mean the Guam seagrasses, it is slightly more impacted by sewage than seagrasses as a whole. Now, what this figure is of a variety of tropical seagrasses and their isotopic values that were collated by Christian et al. In 2013. And so I just took all of the values that they published and then graphed it on our values. So the darker, or I guess the deeper green is the inhalers from Guam, and the brighter green were the inhalers that were already published. And so Guam's seagrasses are slightly on the more impacted, at least in terms of the exposure to nitrogen pollution, than your typical tropical seagrass, including thalassia and a few others. So the impact on the seagrasses appears to be in terms of how extensive there are. And so this was something that people have been worried about. Waycott in 2009 published a paper summarizing all of the available data, something in the order of about 870 studies, looking at the loss of seagrass beds throughout the world. And so they have this value, and they've been declining by about 1.5% per year. A few things to notice. There are no data from the Western Pacific. And they also lament about the fact that there are no data from the Indo-Pacific as well. And so if you're looking for a place to do some work, in terms of seagrasses. And so luckily for us, in 2004, somebody had published a very well-resolved benthic habitat map of entire coastline of Guam. This was a NOAA-funded work. And you can see the little V's here. They're supposed to be seagrasses. So you can actually do the traces and figure out that these are the places where you would find the seagrasses. So we did 2 things. So these are from 2004. This was done in 2014. So we had a decadal change. We're looking at a decadal change in seagrass beds. And so we did 2 things. One is we found more images, WorldView 2 images. These were based on Icona's imagery back in 2002 and 3, around there. The new set of images came out in 2014 from WorldView 2. And so they were used to also do similar kinds of things. And so this is what the WorldView 2 images look like. This is one of the sites on the leeward side. This is an observation deck, so tourists can walk out on this walking bridge. Seagrass beds on either side. They walk out and they can walk down and look at the fishes and stuff like that. But seagrass beds are here. These dark patches are seagrass beds. Everything out there are corals. And so you can use this and trace out the size of these things and compare them to the data from 2004. We had this cockamamie idea that, hey, let's do something really simple, something that could be applied anywhere without having to rely on these very expensive images. And so we decided that the iPhones and the GPSs that they have are pretty good, and so we packed them in a waterproof bag, turned on our GPSs, and we literally walked around each of the seagrass beds. And so this is one of my graduate students walking around. It should be a movie, so. Sorry. Give me 1 second. That should be him walking around. There we go. So we did this for a bunch of seagrass beds. So there's some haliduli in there. There's Lophola in there. So this is not a inhalus bed, because inhalus beds you'd be in like half deep water, and so this is for the deeper sites. So we walked around the perimeter. So a seagrass patch that may be about 1 kilometer long in its totality was about 5 kilometers. Right? So that was kind of interesting. And so it turns out that the difference between the WorldView 2 image tracing and walking around is about 5% in favor of walking around. So if you walk around, the seagrass beds that you measure are about 5% bigger than if you trace it on a computer using WorldView 2 images. I suspect that the ability to really walk around the contours, and literally we were doing this for the entire afternoon with iPhones on our back. And so I think the walking data are probably much better resolved than being able to sort of click on lines using some of these WorldView images. Anyway, 5%, I think that's pretty acceptable. And from those studies, we find that there's been a relatively substantial decline in the seagrass beds, about 33% over the last 10 years, or about 3% per year, which is double what's happening globally. And so this is a map of Guam, and these are the different patches that we walked around. The light green indicates what was there in 2004. The red is what is there now, and blue is what's been added. And so when you see large patches like that, it means that's all gone. And so this red included patches of grass that was there in 2004, but is no longer there in 2014. And so when you see lots of patches of red, like Agana East and Petey, those are all seagrasses that are now gone. And so there are a couple of places where some seagrasses were added, but in most places the seagrass largely disappeared. So this was pretty significant. This is significant because, as we know, seagrasses and the beds that they form are important sinks for carbon, organic carbon. And so this is another recent paper, 2012, where they collated all the data on seagrass carbon estimates, and they came up with a number about 20 petagrams of carbon being stored organically by these seagrass beds. And so they map out all the regions, and just like the previous paper, they lament about the fact that there's no data from the Western Pacific and there's no data from the Indo-Pacific. And so being out in Guam, we were able to fill in a couple of dots, and so these are much happier looking students collecting sea cores, and so we had a PVC pipe that we drove into the substrate, pulled out the core, cleaned it out using a sieve, and then weighed out the biomass, dried it, all those sorts of things. And so the nice thing is that the dried biomass predicts really, really well the organic content, and so we don't have to do this ever again. That is, send it off for organic analysis. We simply dry it and measure it. And what we find is that let me go back 1 second. Inhales has a huge root system, root hairs, rhizomes, and so not surprisingly, most of the biomass that is held in Inhalus beds are below ground, about 8 to 1 in terms of biomass. And so if you scale up and compare the data to other places, we have so these numbers in white indicate the number of studies for each of the regions. We have one for Guam because that's our study, and it is right up there with the productivity of seagrasses in the Mediterranean, which is pretty high. And so we're pretty surprised about that. What this means is that seagrass beds, for which we have very little information, may be very, very substantial sources or the sinks of organic carbon or carbon in general. But we just don't have any estimates to figure that out. In that regard, remote sensing may be a way to figure that out. Right? Looking at WorldView 2 maps and trying to pick out seagrasses in a very coarse way. The last set of questions that we wanted to ask was, has this always been the case? Right? And the answer is probably no. I'm sure things were much better in the past, and so we wanted to gauge the sense of the scale of change that Guam has experienced. And so we used a couple of things. One is corals and the other is foraminiferans to look back in time and to see whether or not these groups, these taxa, have changed both in abundance and taxonomic makeup over the last couple of centuries. Guam, as I said before, is a platform. The reefs reside on a consolidated platform, and so it would be useless drilling down into it because you really couldn't figure out anything. So luckily for us, as these platforms were forming, because it's carbonate, a bunch of holes formed as the water came in and out. And so there are things called holes, coral holes, in a number of places around the island. This is one of our sites. You can't really see the coral hole, but if I zoom up here, right there, whoa, where's it going? Up here, you see that, the light area? That's a coral hole. There are, I think, about 3, as far as we can tell, on the island of Guam. And we picked all 3 of them. That's Agana. This is the second one. This one is much more obvious, right? That's a coral hole, right? And so we went there and carried out some cores. Ordered official aluminum tubes, but this was when there was a strike on the West Coast and nothing was being shipped out to Hawaii or Guam. And so these 8-foot aluminum tubes that we ordered were stuck in Hawaii and they weren't going to make it out to Guam for another couple of months. And so what did we do? Well, we went to Home Depot. And the nice thing is that there's a Home Depot on Guam, right? And so what we ended up doing was getting 8-foot fence posts, the larger diameter ones, and a hammer, a fence post hammer, and we simply drove it into the unconsolidated sediment, holding our breath because we weren't allowed to use scuba for this. And so myself and graduate, we just simply drove it down into the ground. We plugged it, pulled it out, and then because some of the stuff was compacted, we ended up having to take a saw or grinder and cracking open the entire tube. So that was an interesting challenge. And so we have the sediment, because we couldn't gravity feed it because sometimes there were things that were in there that just simply wouldn't fall out by tapping on the sides of the pipe. So we cracked it open and then pushed out 5-centimeter segments and then separated it out, cleaned it, sieved it, pulled out the coral fragments, looked for forams, among other things, to see what the history of this place looked like. Some of the cores went back 400 plus years, and so it gave us some reasonable look into the past of Guam. A couple of things to keep in mind as I present the historical data. About 4,000 years ago, the island of Guam was colonized by people probably from the Philippines, but it wasn't until about the late 1600s when the island was colonized by the Spaniards. And they were there for a long time until 1898. And then shortly thereafter, the U.S. Navy made its home for the Pacific in Guam. And so, in fact, this is largely artificial. It was built so that they can bring in aircraft carriers and battleships into that harbor. And then 1941, Second World War, things changed hands in 1950 when residents of Guam were given U.S. citizenship. Most of the population boom happened after the Second World War, and so there's sort of an exponential growth, especially happening in the last 40, 50 years in Guam. Again, about 160,000 people. So not huge, but on an island that is about 25 miles long and 6 miles wide, that's a good number. So when we sorted out the segments and pulled out the corals by weight, we used coral weight as a proxy for dominance. And so there are a couple of sort of weird assumptions that we had to make. So these coral holes, like traps, things will fall in randomly from around the area. And they will fall in a way that's reflective of the stuff around them. We sort of hypothesized that when there's a huge accumulation of these things, something weird happened, right? Something either killed it off so that lots of fragments were falling in. And if there's a low level of fragment accumulation, then there's sort of the background rate, right? So these are 3 different cores over the last 400 plus years, lots of noise. And so we carried out a P-spline or a penalty spline analysis where we divvy up data according to some arbitrary, these aren't entirely arbitrary, we pick a time and we say, you know, we think that something weird happened here. And so let's compare the slopes of the line of the data before and after that. That's what the P-spline does. And we did 3 things. This is the Spaniard colonization 350 years ago. This is 167 years ago. There are 2 super typhoons that were recorded by the Spaniards. They were so bad they had to write about it. And those were the only 2 that were actually recorded. And so we picked that as a knot for the P-spline analysis. And then this is when this is the end of the Spanish-American War, when U.S. colonization began in earnest. this is sort of anomalous, and so we're going to largely ignore what's happening behind here. Although, ideally, it was really wonderful back then, right? 450 years ago, the coral reefs were luxuriant. It's probably more anomalous than real, and so if you look at the data like back here, this red line represents sort of the baseline accumulation rates of these coral fragments. Things are happening when everything's sort of hunky-dory in the absence of large sort of human influences. Shortly after the typhoons, there's an increase in the number of fragments that are falling into the hole. It peaks and begins to decline, which would suggest that things have gotten better. But the problem is that the numbers are going down. This continued dip probably suggests that the number of fragments or the sources of the fragments are also declining at this time. Right? So our interpretation of the coral core data is that for a long time it's been accumulating at this background rate. It picks up during or shortly after the super typhoons. It peaks and then begins to decline, getting back to normal, but it is also then impacted by human activity. Agana has been developed since the Spaniards. It's been a hub of activity on the island for centuries, including the Philippines that initiated the colonization. And so we have this decline from which it hasn't recovered from. And our guess is that the source of the fragments have also declined. And so numerically, things have changed. But what's also interesting is that compositionally, the reefs have changed. And so if you take the entire core and separate out the dominant members, the acroporids, the postuloporids, they dominate the core for the entire history. If you separate it out 100 years preceding, so everything from 450 years to 100 years ago, it looks pretty similar, right? So for a good part of the core's history, the species composition in Agana was relatively similar. That is, it was dominated by the acroporids and the postuloporids. But if you look at the core for the last 100 years, there's a shift from the branching acroporids to the more robust postuloporids in terms of community dominance. Right? So there's also a species shift happening as well as a numerical change over the last couple of hundred years. These are not that different than the kinds of things that have been happening in the Caribbean. There is a numerical change as well as a shift in the composition. Also, coincidentally, the loss of the branching acroporids from the Caribbean and being replaced by more robust looking corals. The last set of data I'm going to show you are based on the assessment of the baculogypsinid forams. Star sand. And this is the most dominant species that we picked up in the sediments of the cores. And so we did the same thing. We pulled out the sediments and we estimated the abundance by taking out a gram of these things and counting all of the foram in that sample. And these are for two sites, Agana, which is the impacted site, and Shark's Hole, which is farther north, where that clear hole was apparent. And what we see is a couple of things. One is that the abundance of the foram in Agana is way higher than the abundance of forams in Shark's Hole. So there's a numerical difference between the two sites, and Pam has been very helpful trying to figure out why that would be the case. But if you take these data and just turn them into a scatter plot, the trends become a little more obvious. And so this is the number of individuals per gram, years before present, for Agana and for Shark's Hole. And these different colors indicate different cores that we're taking, thrown all together, and you see, at least in Agana, the abundance of Baculogypsina spherulata had been declining sort of systematically over the last number of years, whereas in Shark's Hole, optimistically it has increased in abundance, but at least not has declined over the last 400 or 500 years. Okay. So these data sort of support the hypothesis that there is something going on with the integrity of the coastal ecosystem and the activity of humans on the adjacent lands. So let me summarize by saying that Guam is definitely being impacted by land-based sources of nitrogen. We pick it up in the corals in terms of the Delta 59 signatures. We pick it up in the seagrasses. The corals are being impacted by causing increases in the severity of the disease. We're losing seagrass beds. And we also see that there's been a shift in the composition of the corals that make up these reefs, as well as changes in the abundance of forams, which are interesting, important proxies for the water quality in different places around the island. Okay. The big challenge for Guam is what they're going to do with the influx of people. As I mentioned before, the Pacific Theater is being realigned, and the plan is to reallocate about 11,000 Marines from Okinawa to Guam. But it's not just 11,000 people. There are family members, contractors, and educators, and road builders, all the things that require moving that many people, which could result in an influx of anywhere from 40,000 to 60,000 people in a fairly short order of time. And so that's a concern that is confronting the population on the island, but also portends not very good news for the coastal environment. So with that, I want to thank you for spending the afternoon with me when you could be watching car races. So, thank you.